Thanks for the introduction. I'm a postdoc in the Noble Lab at UW, and today I'll talk about our tool TwinC. It's called TwinC after the snack cake, and it predicts interchromosomal high C contacts in the nucleus from DNA sequences alone. So let's start with high C. It's a next gen sequencing technique that measures pairs of DNA loci that are proximal in the 3D space of the nucleus. The output of the experiment is a contact map where each row and column corresponds to a genomic locus of fixed size, often referred as resolution of a high C map. And we can divide it into an intrachromosomal map for contacts happening within a chromosome, and a much sparser interchromosomal map for contacts happening between chromosomes. So there are several existing methods that do sequence to high C prediction, with Akita and DeepC being the first two models to tackle this task. But these and other methods mostly focus on intrachromosomal contacts because they are more frequent and there's functional relevance, of course, but they ignore interchromosomal contacts, especially because these are very sparse. So there is still evidence that interchromosomal interactions are functional. They are known to assist in formation of transcription factories, assist in active transcription as well as repression. They are known to facilitate the formation of nucleolus, which is the largest organelle in the nucleus, and allow for expression of a single olfactory gene out of hundreds of candidates. So these contacts are functional, and we are interested in learning about their sequence signatures. So the latest in the series of sequence to high C models is the ORCA model that takes as input one sequence of length up to 256 MB and predicts a series of contact maps as an output at different resolutions. This design makes the ORCA model extremely computationally expensive to both train and infer predictions from. But this design, in a way, makes more sense for cis or intrachromosomal contacts because they have structures like domains and loops, which might require larger sequence context. And we also need to account for the genomic distance decay effect. But when we're talking about interchromosomal contacts, we can get rid of a lot of these things and simplify the model considerably because these loci are on different chromosomes and there's no notion of genomic distance anymore. So we propose exactly that in our model TwinC. TwinC takes as input two sequences and predicts whether they are in contact as a supervised classification task. And we deal with the sparsity of interchromosomal contacts using multiple high C experiments from human donors. In this case, we have data from heart, left, and right ventricles. TwinC is a twin convolutional neural network that takes as input 200 KB sequences, and those go through a twin 1D convolutional module with tied parameters. Then those are combined using 2D convolutional modules, and the output is a predicted contact score, which is a score between 0 and 1, representing how likely the two sequences in input are to be in contact. So to get labels for TwinC, we have 10 donors in heart left and right ventricles each. We divide them into two groups. Donor group A, which we use to derive robust positive and negative labels for training. And then label group 2, which we use uh, to get an upper limit of reproducibility given the sparsity and other technical limitations that exist in this data. So we evaluate TwinC using an ROC curve. X-axis is false positive rate, y-axis is true positive rate, and an ideal classifier would be at the top left corner. And as you can see, TwinC is it's in yellow, it's comfortably close to the reproducible upper limit, the blue dot, and it also comfortably beats a GC content based baseline. This is heart left ventricle. We see similar trends in heart right ventricle test set data as well. Even visually, Twinkie's predicted contact map on the right looks similar to the observed contact map on the left. Gray here represents artifacts as well as missing values. Next, we trained a regression version of our model to compare with the state-of-the-art ORCA model. And here you can see that the two models have similar performance. I'm plotting correlation of ORCA's predictions with observed high C on x-axis and Twinkie on y-axis. And the correlation values are close to the diagonal, meaning that the performance of the two models is similar, even though our model is significantly simpler. Specifically, it's 26-fold faster to train Twinkie than the ORCA model, and it requires 1/4 GPU resources. So now that we have this predictive power, we want to assess exactly what this model has learned about interchromosomal contacts. So to do that, we rank our predicted contact scores by we rank our test set predictions by contact score and get two sets, the top 10% and the next 30%. And we do a substitution experiment by substituting attack peaks from the top 10% to the next 30%. So if sequences in the accessible region are important for formation of interchromosomal contacts, which we know they are, and if the model has learned this fact, then this experiment should result in increase in the predicted contact score for this set. That's exactly what we observe. So on x-axis is the predicted contact score before substitution, and on y-axis is the contact score after this substitution. And you can see that all the points are above the diagonal, meaning that the model has indeed learned that these sequences specifically are more likely to be in contact than the ones before. So this is about chromatin accessibility. Next, we wanted to ask specifically which transcription factors are implicated. So we repeated these experiments for 831 transcription factors for humans from the JASPAR database, and defined an enriched set of transcription factors which had significantly higher positive effect on prediction than other transcription factors. And using this set, we next ask if these sets are enriched in interchromosomal contacts, are they also predictive of interchromosomal contacts? So we designed a very simple motif count predictor. And here I'm plotting AUROC of this predictor on the task of predicting interchromosomal contacts. And you can see that the enriched set has significantly higher AUROC on this task compared to other transcription factors. So so Twinkie has indeed learned which transcription factors are enriched in interchromosomal contacts. So in summary, I've shown you that we can do this task of interchromosomal contact prediction from DNA sequence pairs. And despite simpler design and faster runtime, Twinkie's performance is comparable to state-of-the-art ORCA model. It learns about the importance of chromatin accessibility in formation of interchromosomal contacts and finds specific transcription factors that are enriched in these contacts. And in future, we want to see if we can simplify the intrachromosomal part of this model as well, because that would mean more people will be able to apply and use these models. With that, I'd like to thank my postdoc advisor, Bill Noble, our collaborators, as well as members of the Noble Lab. Thank you for listening, and I'll take any questions.
Thank you very much.
Thank you very much for a very interesting talk. Maybe related with also what Bill presented, I think 2 days ago, do you have any lessons about the error, how you should encode the error? Are there any features that you will predict better than others during the training? What's the loss function that you recommend for these kinds of predictions?
Right. So unlike other sequence to high C models that solve a regression task using a mean squared error loss function, we are solving a classification task. So we use binary cross entropy. And the reason for that is interchromosomal contacts are extremely sparse. So you first need a very robust pipeline to figure out which contacts are reproducibly present or absent, and we do that using multiple donors. For these heart ventricles, we have 10 donors, but if you want to repeat this in other tissues, you need at least 3 replicates to be able to reproducibly call positive and negative contacts.
All right, thank you very much. We have to move on.